Dear students, I hope you are all fine with all your activities. As you know that we are discussing about the aspect of embryology. I have circulated notes to you people and I expect that you might have gone through these notes. Just see that today we are dealing with the male reproductive organ of angiospermic plant. It is called as a male gametophyte. Why? Because the male gamete bearing organ, it is called as a male gametophyte. You will also find that this um, male gametophyte, in other words, is also called as an androecium. And this androecium compiled with the help of a large quantity or particular number of individual units called as a stamen. So in a flower, many stamens are present. At a glance, they are called as a androecium. And in that one, anther is present inside, the anther pollen grains are there inside that male gamete is present. So that's why it is called as a male gametophyte. I will show you the structure of this stamen. You will find that this is actually a single stamen. If you see that this stamen, you will find that upper side a particular anther is present. This anther is having a swollen structure and having normally four-lobed structure. Like this one, if you are able to see that, you will find that four-lobed structure is present. And these lobes are joined together with the help of a particular connective that is responsible for joining all these four chambers of anther. And on the adjacent side, there are the sutures. So in this way, this anther is present. Anther is attached with the help of a small rod-like structure called as a filament. This filament is useful for attachment of this anther to flower, as well as it supplies food material to this anther. That's why this anther develops, this anther germinates, and then whatever the pollen grains are inside that one, that develops, and then inner size increases and anther ruptures and pollen grain goes outside. In this way, this stamen is present in case of angiospermic plant. If you go through the transfer section of this particular anther, so you will find that normally it shows such type of structure. If you take the transfer section of the anther and observe under microscope, so you will find that such type of structure is present. You are seeing it on your screen. Now, here you will find that the complete anther is covered with a single layered structure called as a epidermis. Epidermis is a bulliform tissues attached together with the help of their walls and form a complete protective layer, and this protective layer is called as a epidermis. It is a single layered structure. Then beneath this epidermis, endothecium is there. That is a long, thick tissues just present beneath this epidermis and cover the complete anther and provide the protection of all inner tissues, all inner structure. Then you will find that the complete inner part of this is filled with the cortex tissues. You will find this one cortex tissues. They store the food material over here and they supply this food material to this pollen sac or pollen bag. In the central side, as I told you, that a central tissue system is present, which is useful for connection or joining of all these four chambers, and it is called as a connective. If you are able to see this connective, and this connective tissue is hard, thick, and useful for attachment of all these four chamber structure. Then on each chamber, you will find that a particular bag-like structure is present. This bag-like structure is called as a pollen sac. Sac means bag. It is called as a pollen sac because inside it pollen grains are present, and these pollen grains are electron develops and form pollens, or spore mother cells develop and form the spores, and inside which male gamete develops. Now this pollen sac is present and on each chamber, and it is covered by a mid layer, that is a thick layer structure which is present between endothecium and pollen sac, and then this is covered with a thick layer structure which is called as a tapetum. And this tapetum is just to cover the complete structure of this pollen sac. Then towards the marginal side, a thin layer structure is present, and it is called as a stomium. Then this tapetum is somewhat thick, but some delicate part is present towards this pollen sac, and the same one is present towards the anther wall. It is called as tapetum. Tapetum sorry, stomium. Stomium is a thin layer structure. What's happen when this pollen sac matures? It means that pollen grain germinate over there. Then it increases pressure from the inner side, and due to increasing of this pressure, this tapetum ruptures and this pollen grain goes outside. Inside it, the tetrad spore mother cells are present, and that are called as a sporangium. So in this way you will find that sporangium or spores are present in sporangium. Spores are present, and these spores plays important role for various activities, various purpose, as well as we'll find that these pollen grain or pollen mother cells that are the important. In this way, you will find that briefly, if you just go through, so you will find that anther contain a particular tissue structure having a four-chambered structure. It is covered with the epidermis. In beneath the epidermis, hypodermis tissues are present. Then in the central side, connective tissue is present, and on each chamber, pollen sac is present. Pollen sac is covered with the tapetum tissues, and towards the marginal side, stomium is present. Inside this stomium, then we'll find that pollen grain or pollen mother cells are present, and that are having a diploid structure, a diploid condition. All these things you have to study. You will find the complete structure about this epidermal tissues as well as pollen sac. Now, this is just an outline about the stamen, the structure of TS of anther, as well as its inner part. But we'll find that this pollen grain, when goes outside, then it germinate, and this germination is the most important one, and this germination that results in the formation of male gametes. And just I will show you in a simple way. You will find that this pollen grain that goes outside, it is having such type of diploid structure. This pollen mother cell undergoes meiotic division. Meiosis or meiotic division, it is a type of division in which a single cell divide and redivide and form four cells. This single cell is a diploid one, and whatever the four cells formation takes place, that are the haploid one. Now, in this way, the single cells divide and divide, initially form two cells. That cells again goes the division and division process, and then they form four cell structure. In this way, this individual cell structure, which you are all seeing over here, is having then haploid structure, and ultimately in that haploid structure, we will find that microspores are present. And in this way, if you see this structure where you will find this microspores again develop and form this tetrad, वो चौथा जो तो बैक साइड को रहता है. In this way, that microspore formation takes place. Then if we go through the complete individual structure of pollen grain, we'll find that such type of structure is present. Mainly it is having two layer structure, two cells structure, and that two cells structure, uh, uh, two layer structure is called as a pollen grain. The outer layer is somewhat thick, and it is called as a exine. If you see This layer is called as a exine. It is hard, thick, and it is a spiny. If you are able to see this one, these spines are present over here. This spiny layer is useful for attachment of this pollen grain to stigma. It does not slip over there from the stigma. That's why these spines are present. It is a thick layer structure. Then inner complete layer is called as a intine. In this intine, you will find that it is a thick layer structure, fleshy layer structure, and inside it, the nucleus is present. This nucleus plays an important role for the development. But just now I have shown you that one. Then, and this is exine. There are the germ pores are present. Openings are there. This germ pore is actually called as a germination pore. It means that then when this pollen grain start to germinate, it jumps from this, it germinates from this germ pore. This in time enlarges. Ultimately, this exine ruptures over there. And as you find that generative cell is present toward the lower side, in the central side, the tube nucleus is present. It is not the male gametophyte, it germinates. And that results in the formation of male gametophyte. How it germinates? Just I will show you if you are able to see this one. Just see, these are the stages of the germination of this one. This pollen grain which we have seen over there, it germinate, it start to germinate, means this the generative germ cell or generative cell start to enlarge, that division start to continue, and ultimately this in time it enlarges, and these cells goes inside this tube nucleus. This takes place on the stigma after pollination, and this cell, which is called as a generative cell, divide and redivide and form different type of cells, that is called as a tube cells or tube nucleus. That tube nucleus later on present in this pollen tube. This tube is called as a pollen tube. And then this cell divide and redivide and form two male gametes. So after formation of these two male gametes, before fertilization, this tube nucleus and these other cells that also dissolves, and only male gamete remain over there. So remember that one. In case of pollen grains, first they divide and redivide and form four haploid spores. Remember that one. Each haploid spore later on germinate, and each haploid spores form two male gamete. Means from a single pollen grain, we are able to perform four fertilizations. Further, we'll discuss about this one in detail about the formation of fruit. So a single pollen grain is responsible for formation of four fertilization, because they divide and redivide and form four spores, and each spore contain two male gametes. Further, I would like to just inform you that one, in case of angiosperms plants, two male gametes are there, and these two male gametes fuses in a single ovule. That's why it is called as a double fertilization. And this double fertilization is an important characteristic of angiospermic plants. That also we will discuss at the time of fertilization. Now today what we studied the structure of stamen, then structure of pollen grain, then before that one we have gone through the structure of anther, then we have discussed about the microsporogenesis, how this pollen grain divide and redivide and form four cells, and after that one we have discussed about the development of male gamete or male gametophyte. So this is about the microsporogenesis. You have to go through these notes and study it, and if it feels any type of difficulty, so you may ask me. Okay, take care of yourself, and then we'll meet again with female gametophyte. We'll stop here. Thank you.